So good evening everybody. Today we again start our lesson with biotechnology. The second chapter of biotechnology, that is application part. Biotechnology and its application. The subtopic application of biotechnology in health and agriculture. Human insulin and vaccine production, stem cell technology and gene therapy. GMO, that is genetically modified organism, BT crops, transgenic animals, biosafety, biopiracy and patents. So let us move to our first topic. So as usual we know this one. This is our chapter number 12 that shows 11 marks. So we are studying for 11 marks. Now to the subject matter. The introductory part of this chapter, that is biotechnology. As you would have learned from the previous chapter, from the first chapter of biotechnology, that is essentially deals with industrial scale production of biopharmaceuticals and biological using genetically modified microbes, the fungi, plants and animals. The application of biotechnology include the therapeutics, diagnostics, genetically modified crops for agriculture, some processed food, bioremediation, waste treatment and energy production. So the three critical research areas of biotechnology: number one, providing best catalyst in the form of improved organism, usually a microbe or pure enzyme. Second area, creating optimal condition through engineering for a catalyst to act. And the third one, downstream processing. Last day we discussed the downstream processing technology to purify the organic compound, the protein compound. So let us learn how human beings have used biotechnology to improve the quality of human life, especially the particular area of food production and health. So biotechnological application in agriculture. Let us take a look at the three options that can be thought for increasing food production. Number one, agrochemical based agriculture, organic agriculture and genetically engineered crop based agriculture. That's very important. The three thoughts. The Green Revolution succeeded the tripling of the food supply, but yet it is not enough to feed the growing human population. Increased yield have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals. However, the farmers in the developing world, agrochemicals are often too expensive and further increases in yield with existing varieties are not possible using conventional breeding. Is there any alternative path that our understanding of genetics can show so that farmers can obtain maximum yield from the field? Is there a way to minimize the use of the fertilizers and chemicals so that the harmful effects of the environment are reduced? So biotechnology gives this answer. By using the genetically modified crops. This is the solution. So plants, bacteria, fungi and animals whose genes have been altered by manipulation are called GMO, that is genetically modified organism. GM plants have been useful in many ways by genetic modification, such as the crops more tolerant to the abiotic stress, that is cold, drought, salt, heat. This type of stresses are tolerated by these crops. Reduce reliance on chemical pesticide, that is pest resistant crops. Help to reduce post harvest losses, minimize the post harvest losses. Increased efficiency by mineral usage by plants. So that is the prevent that early exhaustion of fertility of the soil. And the enhanced nutritional value of food. You know this one by golden rice. See the diagram. The golden rice, the beta carotene is introduced to enhancement with vitamin A enriched rice. That is the biofortification. You all know that our hidden hunger is the criteria that is fulfilled by the enrichment of the different minerals, vitamins from the beginning. So the peoples of the third world countries are very much benefited by this technology, that by carbohydrate they are getting vitamins and minerals from the beginning. In addition to these uses, genetically modified has been used to create tailor-made plans to supply alternate resources to industries in the form of starches, fuels and pharmaceuticals. Some of the applications of biotechnology in agriculture that you will study in detail, that is the pest resistant plants, which would decrease the amount of pesticide used all over the world. Bt toxin produced by the bacterium Bacillus thuringiensis. Bt toxin gene has been cloned from bacteria and been expressed in plants to provide resistance to insects without the need for insecticides, is effect created to biopesticide. The Bt cotton, Bt corn, rice, tomato, potato and soybean. These are the examples. First with Bt cotton. Some strains of the Bacillus thuringiensis protein that kill certain insects, such as, very important, remember this one, Lepidopterans, that is frequently asked questions in NEET. Lepidopterans, that is the tobacco bollworm and armyworm. Coleopterans, the beetles, and dipterans, the flies and mosquito. So lepidopterans, coleopterans, and dipterans. Bacillus thuringiensis form protein crystal during a particular phase of their growth, and these crystals contain a toxic insecticidal protein. Now the question is, why does the toxin not kill the bacillus? Actually, the Bt toxin protein exists in inactive protoxin, but once the insect digests the inactive toxin, it converted into active form. Remember this one. So protoxin came in contact with the digestive system of insect. It is active. Due to the alkaline pH, because this toxin come in contact with alkaline pH of gut, which solubilize the crystal. So the crystal proteins are getting solubilized by coming in contact with the alkaline pH of intestine. Very important concept. Remember this one. The activated toxin bind to the surface of midgut epithelial cells and create pores that cause cell swelling and lysis of the cell, eventually death of the insect. So how the toxin kill the bacillus? This is the answer. So you can remember the Bt toxin protein crystals are inactive protoxin, come in contact with the alkaline pH of the insect digestive system. They are activated and solubilize the crystals, and this toxin then bind the midgut epithelial cells and create pore on the guts, causing swelling and lysis and eventually death of the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. The choice of genes depend upon the crop and the targeted pests, as most Bt toxin are insect group specific. The toxin is coded by the gene. Remember this one, CRY1AC. There are a number of them. For example, the proteins encoded CRY1AC, CRY2AB control the cotton bollworm, and CRY1AB control the borer insect. So remember this one, there is very important information to remember. So this is the cotton ball and how it destroyed by the bollworm. Look at there. The cotton ball and destroyed by the bollworm, and this one the fully mature cotton ball. Some pest resistant plants. Several nematodes parasite a widely variety of plants and animal, including human beings. A nematode That's the scientific name. Meloidigyne incognitia. Meloidigyne incognitia infect the roots of tobacco plant and causing great reduction in the production of the tobacco. Very important scientific name, the nematode. A novel strategy was adopted to prevent the infestation, which is based on the RNAi, that is RNA interference, takes place in all eukaryotic organisms. Method of cellular defense. This method involves silencing of specific mRNA due to a complementary double-stranded RNA molecule that binds and prevents the translation of the RNA. That is silencing. Very important event. Remember this one. What is silencing? Specific mRNA due to complementary dsRNA molecule that binds to and prevent the translation of the mRNA. The source of the complementary RNA could be form an infection by viruses having RNA genome or mobile genetic element, that is transposons. Transposons. So I think you all recognize the term, that is the question of this year GenPA. Mobile genetic element, the transposons that replicate via an RNA intermediate. Using agrobacterium vectors, nematode-specific genes were introduced into the host plant. This introduction of DNA such that it produced both sense and antisense RNA in the host cell. These two RNAs being complementary to each other formed a dsRNA that initiated RNAi, RNA interference, and thus silenced the specific mRNA of the nematode. This consequence was made that the parasite could survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. That's the pest-resistant plants. See the diagram. The host plant generated dsRNA triggers the protection against the nematode infestation. Roots of the typical control plants, transgenic plant roots, 5 days after deliberate infection of the nematode that protected through novel mechanism that we discussed in the previous page. So biotechnological applications in medicine. Our next topic. The recombinant DNA technology processes have made immense impact on the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce unwanted immunological response as a common in case of similar products isolated from non-human source. At present, about 30 recombinant therapeutics have been approved from human use the world over. In India, 12 of these are presently being marketed. 30 recombinant therapeutics in the world and 12 of them are marketed in India. Best and Bunting in 1921 isolated insulin from the pancreatic islets of dog and demonstrated its effective against diabetes. Best and Bunting. Remember this one. It's important information. Now, how the genetically engineered insulin is produced? It's one of the important questions. You all know this one. Insulin released from the beta cells of the islets of Langerhans of pancreas, used for diabetes, and earlier extracted from the slaughtered cattle and pigs from slaughterhouse. So insulin from an animal source, though caused some patient to develop allergy in the previous stage, another type of reaction to a foreign protein when it is introduced in the early stage. So insulin consists of two short polypeptide chain. One is chain A, another is chain B. And they are linked by disulfide bridges. In mammal, including human, insulin is synthesized by as a prohormone, like proenzyme. Prohormone is also need to process before it become fully mature functional hormone, which is contain extra stage called C peptide. This C peptide is not present in the mature insulin and is removed during maturation into insulin. The main challenge for production of insulin using rDNA techniques and getting insulin assembled into mature form. The company Eli Lilly, the American company, prepared two DNA sequences corresponding the A and B chains of human insulin and introduced them in plasmids of E. coli and produced the insulin chain. The chain A and B are produced separately, extracted and combined by creating disulfide bond between them and form the human insulin. So the concept is clear. Everybody give your response. See the maturation process, the proinsulin. Insulin, that is the A peptide and B peptide, and the C peptide are growing to free. Proinsulin to insulin. This is the flow chart of human insulin production. DNA, human pancreas cell, introduction of the recombinant DNA into the bacterial cell, the recombinant bacterium, from the human producing insulin producing gene, inserted the bacterium, the plasmid DNA, which is cut by the restriction enzyme, and that is the recombinant DNA. Recombinant DNA from recombinant bacterium that multiplying here and producing the human insulin in the fermentation tank. From fermentation tank, the extraction and purification of human insulin, A chain and B chain, C chain is there. Now, here is only A chain and B chain producing the human insulin. So, everybody take a screenshot. It's very easy to show your answer. Human insulin production. So everybody take the screenshot. That's very easy to give the answer in a flow chart and everything in the flow chart. So everybody take the screenshot or not? There is another flow chart showing the production of human growth hormone. See the human cell and bacterial cell. The bacterial chromosome, and that is the plasmid. The gene for human growth hormone and cut by the EcoR1, that is also cut by the EcoR1, forming the sticky end, and they are attached. That is the gene for human growth hormone. Look at here. Then the DNA inserted in the bacterial cell containing human growth hormone. So this is the way how the human growth hormone are produced. Here you see the same type of restriction endonuclease are going to cut in a particular location and forming sticky ends, and then DNA inserted into the bacteria for further human growth hormone production. So it's a very easy way to show all types of biotechnological experiment. You can give this example how the restriction endonuclease are going to function to cut and form the sticky end, and how the DNA are inserted and form a component beneficial for human. So it's a good flow chart for giving any sorts of example in biotechnology. So everybody take the screenshot. It's a very unique one that will help you in giving many answers at a time. Now our next topic, gene therapy. So that is one of the important concept. Gene therapy, see this one. Gene therapy is the collection of methods that allow correction of gene defect. Why we are facing different genetic disease? Because some of the genes are getting defected. So first we diagnose this one in child or in embryo. Here the genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function and compensate the non-functional gene. See the first case of gene therapy, the clinical gene therapy, 1990, not so far. A 4-year-old girl with ADA deficiency, adenosine deaminase. This enzyme is crucial for the immune system of function. The disorder is caused due to deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which the functional ADA is given to the patient by injection. So you have to remember the first case of gene therapy and how the gene therapy is done. But the problem with both these approaches that they are not completely curative in the early days. So as a first step towards the gene therapy, lymphocytes of the blood cell of the patient are grown in a culture outside the body. A functional ADA, complementary DNA, is then introduced into these lymphocytes, which is subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. The gene isolated from the bone marrow cells producing ADA is introduced into the cell in early embryonic stages. It could be a permanent cure. That's the importance of gene therapy. So here a flow chart again for understand the process of gene therapy. Everyone observe this one. It's a very unique flow chart to show the process of gene therapy. The bacterium carrying plasmid with cloned normal human ADA gene. Genetically disabled retrovirus. The T cells with disabled ADA gene isolated from SCID patient. Cloned ADA gene is incorporated into virus. So the retrovirus infect the T cell transfer the ADA gene to cells. Now the cells grown in the culture ensure the ADA gene is active. Now the genetically altered cells are re-implanted to produce adenosine deaminase. So this is the most easiest way to show how the gene therapy will be there. So take a screenshot. That's very important, and not only important, this is the easy way to establish your answer in exam. The process of gene therapy. Now the two approaches to achieve gene therapy are somatic cell and germ-line gene therapy. Very important, very important topic for our turn to. Difference between the somatic cell therapy and germ-line gene therapy. Somatic cell therapy involves the insertion of fully functional and expressible gene into target somatic cell to correct the genetic disease permanently, whereas in germline gene therapy, the introduction of DNA into germ cells which places to the successive generations. So see the difference here. So the therapeutic genes transferred to the somatic cells and these are transferred to the germ cells. That's the difference. Germline gene therapy, somatic cell gene therapy. Introduction of the genes into the somatic cell, the bone marrow, that is the blood cells, skin cell, etc. These are all somatic cells. And here the germ cell, that is the eggs and sperms. The gene are introduced into germ cells. So somatic cell, we all know this one. This is not inherited in the later generation because these are somatic cells. So it can currently cure the patient for a time being, permanently cure of a particular patient, but not inherited in case of the somatic gene therapy. But in germline gene therapy, that is inserted into egg and sperm so that it is heritable and passed into later generation. So note down this one. It's a very important concept and easy to remember. Somatic cell therapy and germline gene therapy. So everybody understood this one regarding somatic cell germ therapy and germline germ therapy. It's an easy concept and easy to remember. So give your response. Is it clear? Okay. Now the molecular diagnosis. Using conventional methods of diagnosis, serum and urine analysis, early detection is not possible. The rDNA technology, PCR, and enzyme-linked immunosorbent assay are the sum of the techniques that serve the purpose of early diagnosis. Remember this one? rDNA technology, rDNA technology, PCR and ELISA. So presence of pathogen, that is bacteria and viruses, is normally suspected only when the pathogen produce the disease symptom. That is common to us. By this time, the concentration of the pathogen is already very high in the body. This is why the symptoms are shown. However, very low concentration of bacteria or virus at that time, when the symptoms of the disease are not yet visible, can be detected by amplification of their nucleic acid and the technique is polymerase chain reaction. So PCR is now routinely used to detect HIV in suspected AIDS patients. So that is done in early way by amplification of the PCR. It is being used to detect the mutation in genes in suspected cancer patients also. It is a powerful technique to identify many genetic disorders. Molecular diagnosis. A single-stranded DNA or RNA tagged with a radioactive molecule that is called probe, is allowed to hybridize to a cDNA, that is complementary DNA, in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have the complementarity with the mutated gene. And the another information of ELISA, that is the principle, ELISA is work on the principle of antigen antibody reaction. Infection by pathogen can be detected by the presence of antigens, that is the proteins and glycoproteins, etc., or by detecting the antibodies synthesized against the pathogen. See some extra flow chart with ELISA. See this one, ELISA is a biochemical procedure discovered by Eva Evangelii and Peter Permanin in 1971 to detect the presence of specific antibodies or antigen in a sample of serum, urine, etc. It is very important diagnostic tool to determine if a person is HIV positive or negative. ELISA is a tool for determining serum antibody concentration, such as antibodies produced in a person infected by the pathogen by HIV, and also for detecting the presence of specific antigens and hormones such as human chorionic gonadotropin, that is HCG. See the flow chart of coding, blocking, detection and readout. So you can take a screenshot for further reference regarding ELISA. This is the ELISA in another way in flowchart. How it is works. the captured antibody, now the targeted antigen. This is the enzyme link detection antibody, and now it is bifurcated into substrate and product. This is the technique of ELISA. Now the transgenic animals. Animals that are their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. That's very important fact. What are called transgenic animals or transgenic plant? Every time that DNA are implemented, DNA manipulated and express the extra gene is called the transgenic. Transgenic rats, transgenic rabbits, pigs, sheep, cow, fish have been produced all through over 95% of all existing transgenic animals are mice. So why are these animals being produced? How can man benefit from such modification of transgenic animals? So now we explore what the reason behind this one. The normal physiology and development and the study of disease, why are these animals being produced? So let us read this matter. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. The study of disease, that is the transgenic models such as cancer, CF, cystic fibrosis, rheumatoid arthritis and Alzheimer's disease. Some biological products. Medicine required to treat certain human diseases can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of the portion of DNA or genes which codes for a particular product as human protein which treat emphysema. Similar attempt has been made for treatment of phenylketonuria and cystic fibrosis. In 1997, the first transgenic cow, remember this one, that's the Rosie, that produced human protein enriched milk. The milk contained human alpha-lactobilin and was nutritionally a more balanced product for human babies than natural cow milk. So that is very important to remember. Vaccine safety are also the another criteria. Safely use the vaccine. In modern day, we all know this one, how the vaccine safety is useful for human being, and before the transgenic animals are used for getting the result either the vaccine is safe or not. Chemical safety testing is also done on the transgenic animals. Whatever the results are found from this, we can take that either it is used on human beings or not, and how the result is beneficial for the mankind. So this type of safety are getting tested on the transgenic animals. So here is an example. How the transgenic milk are produced in goat by transgenic goat. See the human gene and a egg donor. The egg is inserted by micro-injection, and from this the development within this is the host goat, and then the transgenic goat is formed, and from this the milk from the transgenic goat. So this is the example in practical way, the transgenic animals and beneficial for mankind. Some ethical issues. The manipulation of living organism by the human race cannot go for any further without regulation. These are the ethical issues. Some ethical standards are required to evaluate the morality of all human activities that help or harm the living organisms. Going beyond the morality of such issues, the biological significance of such things into important. Genetic modification having unpredictable result when such organisms are introduced into this ecosystem. So these are must be checked in the ethical issues. Therefore, the Indian government, very important, remember this one, GEAC, Genetic Engineering Approval Committee is there, which will make decisions regarding the validity of GM research and the safety, introducing any GMO for public services. The modification usage of living organism for public service as food and medicine sources has also created problem with patents granted for the same. Now the biopiracy. It's a very important topic. Biopiracy is the term used to refer to use the bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Most of the industrialized nation and rich financially but poor in biodiversity and traditional knowledge. In contrast, the developed and the underdeveloped world, rich in biodiversity and traditional knowledge related with bioresearch. So this traditional knowledge related to bioresources can exploit it to develop modern application and can also used to save time, effort, expenditure during their commercialization. There has been growing realization in the injustice, inadequate compensation, and benefit sharing between developed and developing countries. Therefore, one nation are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment in the Indian Patent Bill that takes such issue into consideration, including patent terms, emergency provision, research and development in acting. So everybody understood this one, the biopiracy and its effect. So there is the growing public anger that certain companies are being granted patents for their products and technologies that make use of genetic materials, plants and other biological resources that have been long identified, developed, used by the farmers of indigenous people in specific region and country. Rice is an important food grain. We all know this one, which goes back thousands of years in Asia's agricultural history, and there are 2 lakh varieties of rice in India alone. This diversity of rice, one of the richest in the world. Basmati rice, distinct from its unique aroma and flavor. 27 varieties of basmati grown in India. There is reference to basmati in ancient text. In 1997, an American company got patent rights on basmati rice through US Patent and Trademark Office. This allowed the company to sell the new variety of basmati in US and abroad. The new variety has actually been derived from Indian farmer varieties, because we have 27 varieties of basmati. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or a novelty. The patent extends to functional equivalent, implying that other people selling basmati rice would be restricted by the patent. Several attempts have been made to patent use, products and processes based on Indian traditional herbal medicine, that is turmeric, neem. So, lot of cases regarding these ethical issues are all over going. Not only India, this is going in all over the world. So we are almost completed the different parts of this chapter. Here is a mind map for easy revise of this chapter. What is biopatent, biopiracy, what is GESE and ethical issues. Now, what is the utility of the transgenic animals? These are the application group. In agriculture, the biofertilizer, GMO, micropropagation, biopesticide, gene therapy, genetically engineered insulin, that is Humulin, molecular diagnosis like PCR, ELISA, and pest resistance by BT cotton, BT toxin, and RNAi by pest resistance variety. So that chart will help you to recover all the components from this chapter. So from this, we end our lesson today, and we only remaining two chapter to complete this term 2 chapter of CBSE term 2. So everybody, I think you all benefited by the revision of these chapters. Till then, bye everybody. Good night.